Hello, guys. Today the topic is Enterococcus, and I will try to make this topic the easiest I can make. Sheet for this topic, starting with the name of this bacteria, which is Enterococci. So, entero means intestine. So, this cocci is the bacteria which resides in intestine. As it resides in intestine, we can infer few things from this. First is that it can survive in an environment which has a alkaline pH, as we know that the small intestine has a pH of 9.6. So, this can survive in a pH which is alkaline. Also, it can survive in the presence of bile. The Enterococcus has the ability to grow in presence of 40% of bile. It can also grow in presence of 6.5% of sodium chloride, and it has a unique ability amongst the bacteria that it can grow as the temperature of 10 degrees Celsius. Now, coming to the virulence factors of this bacteria has four virulence factors. So, these are cytolysine, extracellular surface protein, lipotecoic acid antigen, coccolysine, and pheromones. Cytolysins, as the name suggests, cyto means the cell, and lysine means they are causing the lysis of the cell. Now we come to the lab diagnosis of this bacteria. So this bacteria is a diplococci, means that it is arranged in pairs. This is a gram positive cocci. On culture, the characteristic are on blood agar, this shows no hemolysis, which is also known as gamma hemolysis. Then on McConkey agar, it shows minute magenta pink colonies. As I told you that it can grow in presence of bile. So it hydrolyzes the bile of bile esculin and changes the color to black color. This Enterococcus, it consists of two kind of species. One is Enterococcus faecalis, the second is Enterococcus faecium. It is very important to differentiate between these two bacteria. So to make this task easy, I will suggest few tricks through which we can remember. Hello, welcome back. So I was talking about how to differentiate Enterococcus faecalis and Enterococcus faecium. Two basic differences. First, which we can remember very easily. The first difference is amongst these two, the name of faecium is larger. So, this requires the testing of more drugs because it is showing multiple drug resistance. So, more MDR is being shown in faecium. The first difference. And fecalis ends with less, so it shows less drug resistance. In the biochemical properties, especially the fermentation of sugar, we test for four sugars: arabinose, mannose, pyruvate, and sorbitol. Amongst these four sugars, the name of arabinose is the longest. It has the largest number of letters amongst all these. Similarly, faecium has more number of letters. So, arabinose is the only sugar which is fermented amongst all these. The rest three are non-fermented by faecium. And this is the opposite for faecalis. Faecalis ferments mannose, pyruvate, and sorbitol, but it does not ferment arabinose. So, these are two basic differences between these two. So, hope I was able to make this topic easier for whoever is listening.